So, hello everyone. We are once again in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. And today is a first for us because after more than 50 interviews, is the first time we are interviewing someone with experience in fossils and in this very, very interesting type of mycorrhizal research. So we have the honor to have with us to Christine Strullu-Derrien. I'm hoping that I'm pronouncing your last name
Yes,
correctly.
it's correct.
And well, Christine is very well known in our field, and I myself always cite a 2018 paper which show the oldest fossil of AMF or endomycorrhizas. And they recently published a very nice new phytologist paper showing a 407 million fossil in Scotland. In the description of the video, you will find the title and the link for this paper in New Phytologist. I wanted to start asking you what is the context of this study? This is a big grant that you have, or is a maybe a postdoc or a student work? What is the context of this study?
In fact, I am working as an independent researcher, so it's difficult for me to have a PhD student or even postdoc. So it's mostly my work, I will say. And this is part of a project. I received a grant last year from the Fondation de France. And in this Fondation de France, especially the foundation Professor Atanas Sakas. So it's a 3 year grant.
Yes.
So that support my work on fossil fungi.
Mhm.
So the idea is to continue what I've done before, to work on the origin and early evolution of fungi associated with plants, and not only fungi, other microorganisms like mycete or everything I can find, but always in a plant environment.
In a plant environment. And in that sense, for people who have never heard about microbiome fossils or fossil, plant microorganism fossils, and in general mycorrhizal fossils, how common or not common are these kind of fossils, and how difficult are they to find and to detect?
They are not common at all. The problem is you have to find exceptional preservation to be able to, to show fossils because they are microorganisms, so they are very small scale.
Mhm.
And there are two main issues. The first is this, you have to find this type of preservation. And the second time is there is a lack of terrestrial sediment for a long period of time. For the period we suspect they arrive on land with the plant. And so it's why it's rare to find the material to work on them. And so the best one and is unique of this 407 million years old. For a chart is from Scotland, from the village of Riney, but there are two deposits in this. There is what is called the famous Riney chart, but there is another chart that is called the Windy Field chart that has been more recently discovered and is far less well studied than the Riney chart.
Wow. And I remember seeing in the literature that maybe the oldest spores are like 460 million years, if I am correct. But the Rainy chart and this second chart, what you are showing is the structures and particularly arbuscules. So how would you explain this difference between the oldest spores, right? And then the oldest mycorrhizal structures.
But the oldest spores, 460 million years, are controversial.
Okay.
So the spore has not been found as associated with plant
Uh,
first. Is, and um, and also it is been found in dolomite and in marine dolomite,
ah,
and that is a very porous sediment. So many geologists, paleontologists consider that it can be contamination.
oh, thank you.
So yeah, that's the big issue.
Yeah.
And so I discussed this recently with John Taylor. I met in London last summer. This work has been done by Redeker when he was in Berkeley. So the slide are in Berkeley. So I plan to go, well, maybe next year, to try to see if we can find a way of proving that they are contamination or not.
Oh,
Because for the moment, most of the people who know this type of rock consider that it cannot be possible that they were fossilized in this type of rocks.
oh, that's so nice to know because yeah, I didn't know this. Yeah.
No, it's not well known. It's known in our community, but not well known out of no. And so the only really non-controversial are from the Rani chart. So before we don't know. We suspect that it should be there before, but for the moment there is no record that we can be confident on.
And that brings me to my third question, which is precisely about the rainy shirt. How long have you been studying this specific site, and what have been your main findings?
Yeah, I got a Marie Curie grant to do a postdoc in London in 2012, and that was supposed to be for 2 years to work on the Ranicha. I'm still working on the Ranicha.
Nice.
so, so more, and I think I will work along still, because now with this windy filter that offer another possibility, I borrow some piece of rocks from the museum in Edinburgh, and since section have been made in London, and so now I have this material that I can work on, and I have, I think, 30 thin section or something like that. So it will, for the moment, I'm focusing on that. And also, I try to move a little in the younger sediment, in the Carboniferous sediment, because in the Carboniferous, you have shared in some part, in some places, but you have also what is called cold balls. So they like balls. And in this material, the plants and the microorganism are silicified like the same way in the chart. So it's another possibility, another type of material to work on.
And I think you already kind of mentioned some of the challenges, but I was wondering about the technical challenges of studying this kind of material.
Uh, um, the, the, so when I did the postdoc, it was to work on the historical collection, the collection that has been made in the beginning of the 20th century. So, but they were perfectly prepared, so you can work on them in bright field microscopy. But the problem in bright field microscopy, you have only the 2D. So that's can be an issue to interpret. And so in 2000, I think it's 2013 or something like that, I tried to use confocal microscopy. And in that way, in the confocal microscopy, you can get the 3D with you can have a stack of image, and then with imaging software, you can reconstruct the organism in 3D. But for example, then I wanted to do some chemical analysis, but on the historical collection, I cannot because they are covered with cover slip, so you cannot have access directly to the material. So now is why I try to ask to prepare the scene section without cover slip. They are very well polished, and then you can do more on this. The other issue is they are historical collection, so sometimes you cannot borrow them from the museum where they are because they don't like, they agree that you come on site, but not, you cannot borrow them in many places. That is another problem sometimes. The other issue is that they are rare. They are rare because charts are not a type of geological material that is common. So these are the main issues.
Excellent. Yes, I thank you. I imagine that those were some of the main challenges. So now let's talk about this recent paper. I don't know if you can share your screen to show the article, and also you mentioned that here you use confocal microscopy, right?
Yeah, yes. So how can I share this? Cannot share this. I cannot find where to share it. Oh, I cannot find where partager. Maybe that. Maybe that.
It should be a green kind of button.
Yes, I had the green, but they say that only one can partager. If I go that way.
So yes, this very nice article, an arbuscular mycorrhiza from the 407 million year old wine filter identified through advanced fluorescence and Raman imaging. So can you explain a little bit what are those two techniques in the title, fluorescence and Raman imaging?
Yes, so the fluorescence imaging techniques are the confocal microscopy and the film microscopy, fluorescence lifetime imaging microscopy. And the Raman are the chemical analysis. So first for on this material, the first thing has been to have the thin section prepared, and then to look at the material in a bright field microscopy. So that are the two images on the left part. And so we were able to show with this technique that you have this line, this brown line, that is the colonization by the fungus, and the zone where the arbuscules form. And then you have also spore in wide field microscopy. Then we decided to go to confocal microscopy to be able to have the structure in higher resolution. You get far more details. We have the 3D, but you also had far more details when you go to the confocal microscopy. So we were able, for example, to show the H shape ramification, that in fact what is called sometimes septation is not septation, because when you look inside there is nothing inside, it's just a checking of the hyphae. We were able to show the shape of the spores, the shape of the vesicles, and also that the spore, you have this septum, the base of the hyphae. And then we look at the arbuscule. So the arbuscule first is on the left is in bright field microscopy. Then it is in confocal microscopy, and the last one is in film. So you can see immediately that how the technique show better, in a better resolution, the arbuscules.
I mean,
So,
no, sorry,
so
go ahead.
the
Go ahead.
So confocal already gave a good result, but FLIM is even better because with FLIM you can separate the arbuscule from the content of the cell, and that was the other technique. Yes. So here you have on the left you have the arbuscule inside of the cells. So what is green is the plant cell. What is blue is the arbuscule. So then you can separate the two, and you have the arbuscule really in focus. And then we also observed that this plant, it was known, but not really well shown, that the vascular system of the plant that you have here, this plant is I forgot to say that this plant is aglaophyton. So this plant has a vascular strand, the tracheids in the middle of the axis. They are particular. This has been shown not very well previously, but the confocal help really to show that, in fact, the tracheids contain inside small vesicles, very small vesicles. This is not a taphonomic process. This is a part of the tracheids. And so we FLIM analysis. Surprisingly, we found that they have the same. They have the same fluorescence lifetime, and so some are fungal and some are plant components. So why we get the same signal from a fungus and from a plant component? So the idea was to look at these two structure using Raman spectrometry, and the Raman showed that in fact you see the similar signal for the two of them, and so that means that the structure have the same the similar chemical composition. So in fact, what happened for this material? During geological time, the diagnosis process transform the arbuscules and the tracheid component in the same chemical component. So we lose the original component, chemical component, and so in the future we will try to go to the molecular to try from this component to see what more. We will try to see if we can go further, if we can go to very high, maybe synchrotron, I don't know, if we can get small differences in the two to be able to say, okay, now you have a similar chemical component, but there was still some little molecule that helped to say that it was fungus or a plant.
That's great, and it was a very nice explanation of your main findings. Thank you so much for being so succinct. It was very clear. The photos, the arbuscule photos are beautiful. I mean, they are even better than current living
Yes.
living arbuscules.
Yes, I cut many living things, roots, and I say, oh, these are better.
Yeah, yeah, absolutely, absolutely.
But it's not surprising because it's fixed in a certain
Yeah,
time.
yeah.
So that's why. But you cannot repeat. I mean, the answer the issue with the slide is you cannot make a series of sections. You have this. Now with the block, I made a series, but originally the problem also with this historical collection is that they did not say this is 1, 2, 3, 4, 5. They did not say that you have series. So it's very rare that you find two following slides, very rare.
Huh.
But now with the
And
new material, I try to make the following section.
and for the from this figure that you are showing at the moment, it's amazing. So if I understood correctly, the chemical composition changed over time, right?
Yeah, yes.
And you can infer this from the figure. That's quite,
It's,
quite
this is, you see, this one is very EO2. You have the arbuscules in green and the vesicle of the vascular strand in red, if I don't say the thing in the wrong way.
yeah.
No, it's the wrong way. It's
It's,
the,
yeah,
the arbuscule in red and the vesicles of the vascular strand in gray.
yeah.
But it's very, you see it very similar, both of the.
So I have two remaining questions. If you could summarize this research like in a takeaway message or in a takeaway conclusion, what's kind of the main conclusion of this study?
In fact, what is interesting in this study is that we have been able to show that aglaophyton is colonized by two type of fungi depending of the environment,
Oh,
because you have the subvascular mycorrhiza in the rainy chart, and now we have showed that we have also in the windy field chart, where the environment is not exactly the same.
wow.
You have different arthropods, for example, and the windy field chart is considered a little more wet. So
Hmm.
you have ponds, and so like in modern den, you know you can have a plant colonized by the same but different fungus
Hmm.
depending of the environment where they live. So that was an important finding of this, and also this combination of techniques showed that you can use this now in the future for other to find other mycorrhiza or other type of fungal association, because in the Rhinicher now we have the mycorrhiza are not the mycorrhizal fungi are not the most common fungi in the
Oh,
chart. No, they are rare. They are rare, very rare,
also
very rare.
the most common fungi are
The
agaricum,
saprophyte, the
saprophyte.
saprophyte,
Oh,
the
wow,
saprophyte.
wow.
A lot, and you have a lot of zoosporic fungi, the Blastocladio or Chytridium mycota.
Mm.
And you have also some ascomycetes. Now, last year, no, 2 years ago, I published a paper describing the earliest pathogen, that is an ascomycete. And so the mycorrhiza are rare. They have been found in two plants now, only two plants. In another plant, we can see a fungal colonization, but we have never been able to show the arbuscules. So until we don't have the arbuscule, we need to have the arbuscule to show that the association was active. So
Yeah, was
yeah.
functional. Yeah.
And
Uh,
also, I found some axes that are not colonized.
wow.
So I am wondering if the association is not like, for example, you have a isoetes nowadays. When the plant is submerged, the fungus is not there. And
Mm.
when the plant out of the water, you have the mycorrhizal association. So is it a kind of thing like that? I don't know. I
Oh,
don't know.
that's that's so yeah, that's so interesting. And
And also the important thing, I gave a seminar in Uppsala two months ago, and it was interesting to discuss with the colleagues because they were working on living material. And in this plant, the colonization is not on the roots because this plant have no roots.
oh,
The colonization is at the base of the stem.
oh wow.
Yeah, yeah. and people don't realize that. Yeah. And the strange thing is this that this plant have rhizoids. They have no roots, but they have rhizoids. And the colonization is not from by the rhizoids.
Huh.
So it's between the outer layer, you have the outer layer between cells, but it's not the rhizoid are not colonized. It's very surprising, and I found beautifully preserved rhizoid, but the fungus is not there,
Wow. Well, it
and
might.
so it's more like what you have in the liverworts,
Mhm.
onwards. It's the time of where you have the chlorophyll whose part colonized. And but why is not by the rhizoid? Because in this plant it's by the rhizoid. So that is an open question.
Well, it's my first time hearing this and many other things. Thank you so much because I'm really learning a lot. Um, the last question is in this kind of research, what are the next steps for you?
I continue to. I cannot put the Rhine chart on side.
Yeah.
I continue to work on the Rhine chart, on the windy feature now with the new series, and I am also working on chart from the Carboniferous of France. We just have a paper describing a new omicete that is just accepted.
Nice.
So, and sometime I collaborated with Ludovic Lerenard on a paper about epiphyllous fungi. That is also an area that I would like to investigate because we don't know when they really originate. And they are quite common in the Mesozoic, but they should have been there before because we have lived before. And also the ectomycorrhiza, because I want to find the oldest ectomycorrhiza.
Wow, that's so exciting.
Because the ectomycorrhiza, the fossil record is around 50 million, but we can be sure that they were there before.
Yeah.
So I try to look at Triassic material and material where you supposedly be able to find them.
Well, that's absolutely exciting. I agree that they are much older than that. It
Yeah, yeah.
seems. So that's very exciting to hear, and thank you so much for your time. And sorry for my connection problems, but I think that it will be get out nicely. And look very much looking forward your future research. It's always very nice to read your papers and to update the slides with my teaching slides with what you have finding because it's always so exciting to hear this kind of research.
Thank you very much. I'm very happy that to be able to show to our community because it's not a field we well know. And
Yeah,
thank you for this invitation to participate to this talk.
thank you.